The Codebreaker, the story of Dr. Jennifer Doudna. In the small town of Hilo, Hawaii, surrounded by volcanoes, waves, and lush green forests, a young girl, but this new power brought ethical challenges. Should we edit human embryos? What about designer babies? Could CRISPR be misused? Jennifer Doudna was one of the first scientists to sound the alarm. She organized global meetings to discuss the moral implications of gene editing, advocating for strong ethical guidelines. In a now famous moment, she described waking from a nightmare in which Adolf Hitler approached her and asked to learn about CRISPR. The dream haunted her, a symbol of how science, if used wrongly, could bring harm rather than healing. Despite the ethical dilemmas, Doudna's work continued to blossom. In 2020, she and Charpentier were awarded the Nobel Prize in Chemistry, making them the first two women to share a Nobel in the sciences without a man. It was a triumph not only for gene editing but also for women in STEM. Doudna became a household name, a real-life codebreaker, unlocking the secrets of life. As the COVID-19 pandemic gripped the world, CRISPR was once again in the spotlight. Doudna's lab at the University of California, Berkeley, pivoted to develop rapid CRISPR-based testing for the virus. While others fought the pandemic on the front lines, she and her team fought it in the lab, proving once again that science saves lives. Dr. Jennifer Doudna's story is more than the tale of a brilliant scientist. It is the story of a girl who defied stereotypes, followed her curiosity, and helped unlock the very language of life. She turned a molecule into a tool into a cure, and a cure into hope. In a world that often underestimates the quiet thinkers, she became a symbol of what is possible when science, ethics, and courage come together. The Codebreaker, the story of Dr. Jennifer Doudna. In the small town of Hilo, Hawaii, surrounded by volcanoes, waves, and lush green forests, a young girl named Jennifer Doudna grew up asking questions. Born in 1964, she was curious, quiet, and thoughtful. She loved reading books, hiking through nature, and wondering about how life worked, not just the way things moved, but why they existed in the first place. Her father, a literature professor, gave her a copy of The Double Helix by James Watson when she was in 6th grade. That book, the dramatic tale of how scientists discovered the structure of DNA, changed her life. Jennifer was hooked. The idea that something as small as a molecule could define the fate of all living things fascinated her. But being a girl in science wasn't easy. In high school, when she told her guidance counselor she wanted to become a scientist, the counselor looked surprised and said, "That's not a job for a girl." Jennifer didn't argue. She just worked harder. She earned a degree in biochemistry from Pomona College and went on to complete her PhD at Harvard, under the mentorship of Jack Szostak, one of the leading molecular biologists of the time. In the male-dominated world of academic science, Jennifer pushed through every barrier with grace and resilience. Her early work was focused on RNA, the lesser-known sibling of DNA. While DNA carries the instructions for life, RNA is the molecule that helps carry out those instructions. Jennifer believed RNA was more than just a helper. She believed it had secrets waiting to be discovered. This belief led her on a path that would one day change the world. In the early 2000s, Doudna became fascinated by a strange immune system used by bacteria to fight off viruses. When a virus attacks a bacterium, the bacterium stores pieces of the virus's DNA in special segments of its own genome, called CRISPR, clustered regularly interspaced short palindromic repeats. This genetic memory lets bacteria recognize and destroy viruses if they attack again, like a biological mugshot system. Alongside this, bacteria use an enzyme called Cas9 to cut the viral DNA, neutralizing it. This natural system had evolved over billions of years. But scientists began to wonder, could CRISPR-Cas9 be used not just in bacteria, but in humans? Doudna, working with French scientist Emmanuel Charpentier, embarked on a groundbreaking collaboration. In 2012, the two women published a paper that would go down as one of the most important in modern biology. They showed that CRISPR-Cas9 could be programmed, like software, to cut DNA at precise locations. Suddenly, Editing the genome of any organism became fast, cheap, and accurate. It was a revolution. This discovery opened the door to treating genetic diseases like sickle cell anemia, muscular dystrophy, and even certain cancers. Crops could be made more resilient. Viruses could be defeated by rewriting their genetic code. Life itself could be edited.